Okay, so here we go. We've been talking this whole time about Mendel and Mendelian laws with regards to genetics and dominance and recessiveness, but as some of you figured out while doing the lab we were working on, there are some other scenarios that can occur with regards to genetics and heredity, and there are exceptions to what Mendel did. This up here actually isn't one, and maybe we'll talk more about that a little bit later. All right, so Mendel, he did some great work, and he happened to pick peas to work on, and he was so lucky that peas happened to have traits and genetics that are dominant and recessive and very easy and basic to understand. And that actually helped Mendel out a lot in being able to successfully determine sort of the causes of heredity. But later on, as people started to look at other stuff other than peas, we've discovered that there are exceptions. Okay, first off, in determining gender, peas have both male and female parts on each flower. So there's not a difference in gender when it comes to peas, but there are a lot of organisms that have separate sexes. In humans, the 23rd pair of chromosomes is what determines gender, and these chromosomes are known as the sex chromosomes. Females have an XX genotype, so they have two X chromosomes. Males have an XY genotype, so they have an X chromosome and they have a Y chromosome. If you look at a picture of all of the chromosomes that we have in one cell, you can see that there are 23 pairs, okay? And almost all of them in this picture are actual homologous pairs where they have the same sequence of genes on both of them. In this case, though, the sex chromosomes for this individual are an X and a Y chromosome, so it is not exactly a homologous pair. It's it has one X and one Y. It's heterozygous for that last pair of chromosomes if you're a guy. All right, so to determine, when you're looking at determining or making predictions about sex and determination in a person, females produce eggs with only X chromosomes because that's all females have. They have two X chromosomes. They can only give an X chromosome to the baby. Males, however, produce sperm that have either an X or a Y because the genotype for a male is XY, so they can either give an X chromosome or a Y chromosome to the baby. So what is the probability of having a girl or boy child? Well, if we go through the Punnett square here, if the two X's get together, one from the male, one from the mom, that's XX. And you can see overall that what turns out is you have a 2-to-2, or if you reduce it 1-to-1, which is a 50% chance each of actually having a boy or a girl. All right? Does anyone have any idea why it's impossible to have a 3-to-1 ratio when it comes to this? If you'll recall, the only way we see 3-to-1 ratios is if you have two heterozygous individuals that are mating. Okay, well, females are always homozygous because they have to have two X's. Males are our only heterozygous situation, and two males cannot have a baby. So that means that's the reason why we'll never get a 3-to-1 ratio. It will always be 50-50 every time a male and female mate of whether or not they have a boy or a girl. All right, another exception to Mendel's rule is something called incomplete dominance. And sometimes an allele is not completely dominant over another trait, and you get that blending or that sharing of influences that a lot of people at the time of Mendel believed to happen, and as we later found out, it actually does happen in some instances. For example, red snapdragons. If a red snapdragon flower is crossed with a white snapdragon, that F1 generation, if we're dealing with true breeders, that F1 generation will all be pink, not red or white, okay? It's sort of in between. And then if you take that F1 generation and have them self-pollinate, you get an F2 generation that is 25% red, 50% pink, and 25% white. All right, so even though the F1 generation has that pink, its genotype is heterozygous, and it therefore has the alleles for both red and white, and then they reappear in that second generation. So here's the cross, okay, and there's two ways to do this. This example is using two different letters to show that neither one is dominant over the other one. And so RR is red, WW is white, but WR or RW is pink. And so if we take two WRs and cross them, we get 25% white, 50% pink, and 25% red. That is incomplete dominance. You can also do Punnett squares like this guy over here, where you show the genetic kind of place represented by C for color, and then you have a superscript for red and a superscript for white. You can do it this way. Different scientists do it different ways. Whatever you like works for me. Same situation though. Red with homozygous red alleles. True breeder white with homozygous white alleles crossing together forms the pink heterozygous. And then the heterozygous crosses produces 25% to 50% to 25%, or 1 to 2 to 1. Okay? I kind of like the sideways Punnett squares, diamonds. All right, codominance. Sometimes both alleles will express themselves independently, and both phenotypes will be visible in the individual, and that's what codominance is. So it's a shared dominance. Both appear, and you can see that both white and red are in this flower. Okay, so codominance, just by definition, is an organism that has two different alleles of a gene. If it has two different alleles for a gene, it shows both of those phenotypes at the same time. They don't blend, they both show up. So here's an example. If you have a black cat crossing with a tan cat, you'll get a cat that has both black hair and brown hair all together, and you get that sort of tabby color. All right? And it occurs with chickens. If you get a black and a white chicken together, you'll get both black and white feathers appearing checkered in the individual. So that's kind of cool. This all got the question. In the F2 generation over here, what is the probability of having a Rhone horse? Remember, Rhone is the mix between the white and the red, or the chestnut. Most of you should say 2 out of 4, which is 50%. Okay. Sometimes, here's another exception, there are more than 2 alleles for a particular gene. Blood type is an example of this, where there are 3 alleles involved, the A, the B, and the O alleles. And depending on what you have, that determines your blood type. All right, so here are the various genotypes you can have for blood type. You can be A and O together, which makes you A. You can be homozygous A, AA. You can be heterozygous BO, but that will make you B blood group. Or you can be homozygous B. You can be heterozygous AB, which will give you AB blood type, or you can be homozygous O, which gives you O blood type. Basically, O is an absence of a particular protein on the surface membrane of the blood cell. And you can see that over here. Like A type, you have A's all over the surface of the blood cell. With AB, you have both the A and the B all over the surface. So it's really actually a codominance kind of situation for A and B, because both of them are expressed. And then O has nothing, or at least not any recognizable surface proteins that are important. Okay, so that's multiple alleles. You can also see over here, I think I show on the next slide, if you were going to do a cross. So if you had an individual that was heterozygous BO with a heterozygous AO and cross them,
you could get all the blood types. That's kind of fun. So if this A and this B get together, you would have blood type AB. If the A and the O, you would be A. If the B and the O get together, you would be B type blood. And if the two O's got together, you would be O type blood. And there you have it. So I wonder if mosquitoes really do favor a particular type of blood. Be interesting to test that. All right, another exception. There's one gene, many traits, where a single gene can influence more than one trait in an organism or can be linked to other traits in an organism. The example that's being pictured there is in large cats, fur color, eye color, and hearing are all linked together. So if you get a cat, a white, in this case a white tiger, and it has blue eyes, I mean it'll generally end up having blue eyes, and it will also unfortunately be most likely deaf. It's sort of just a combo that happens with just this one gene influencing several different things. All right. This is also known as pleiotropy. That's a scientific term, pleiotropy. Another pleiotropy example is a disorder called Marfan Syndrome. And it's one gene that codes for a protein that influences connective tissue development. And connective tissue is anything from that'll be in your joints, like cartilage and stuff. It also influences blood. And basically you end up having problems like, lanky skeleton, leaky heart valves because the connective tissue in the heart valves isn't working properly, weakened blood vessels, lens displacement in the eye. And I don't know if you can see that in this picture, but see this curvature right here? That's the lens. The lens is supposed to sit in the middle so it focuses properly, but the lens isn't, the connective tissue that holds it in place isn't working right, so it's kind of fallen to the side. A lot of historians and scientific historians believe that Lincoln may have had this problem just due to his overall descriptions of his body and problems, ailments that he had. So there you go. Next exception. Sometimes there are several genes that can control a particular trait. So in the case of, say, parakeet feathers, human skin, human eye color, it's not just one gene that's determining what the phenotype is. It's actually several in some cases. Sometimes it's just 2, but sometimes it's 3, and sometimes it may be as many as 6. And this is known as polygenic inheritance, polygenes, right? More than one genes. And parakeets are pretty cute. Budgies. Okay, so here is an example of a cross showing 2 genes and the alleles involved to create the various color patterns in the parakeets. All right. This cross is known as a dihybrid cross. You do not have to know it, okay? But because you have 2 genes that are linked together and involved, there's a lot more possibilities of combinations that you can get, all right? So like if this is one parent and they're both heterozygous, right? So big B, big C, little c, all right? So this B and this C could be part of the allele that it gives to the baby. But this B could also get together with the little c and be something that it gives to the baby. The little b could get with the big C, or the little b could get with the little c. Okay, so these are all the possible alleles that the parents can give, which then gives you this huge combination of it's basically 9 to 3 to 3 to 1 ratio of different color patterns that you can get in parakeets. Again, you will not need to know how to set up a dihybrid cross, but I just wanted to show it to you, okay? That's how you get a white parakeet. With skin color in humans, there are at least 3 genes that control it, and some scientists think there may be as many as 6. I think your book says maybe 4. And when you have that many genes involved, there is a huge spectrum of different variabilities that you can have. And you can see it here. You've got, these are all this, well, these 3 and these 3 are the same. This one's by itself really, really dark. Then you've got this selection that are sort of similar. I mean, it's a lot going on, okay? So that's why when you look at people, you see such more of it's more of a range of smooth changes in color variations between people, and it's not as obvious that you're this for skin color and you're this for skin color. Okay, it's all much more varied across the spectrum. And there's 64 different possibilities from this cross of 3-on-3 genes. All right. Eye color is another one that is controlled by more than one gene. There's at least 2 genes that cover it, and there's probably more. This one is showing another dihybrid cross, and you can have these variations. They're not really showing the green color. They're showing it kind of partially there. This little thing might be fun. You may want to pause and type this in, because what you can do with this particular website, I'm not sure it's going to let me do this or not, but all right. Another thing that can influence your phenotypes is the environment, funnily enough. I mean, it's not just genetics. There's several instances of environmental factors influencing physical features in development that otherwise would be just controlled genetically. Examples are things like temperature with a lot of different reptiles. If the temperatures are in a certain range during the egg development, it can cause the babies to become one sex versus the other. In the example I have here, the higher temperatures tend to produce more males than the lower temperatures, and then there's a medium that tends to be more balanced. With some turtles, it's the opposite, where the higher temperatures produce the girls, and the lower temperatures produce the boys. It's kind of funny how it does that. The other thing with people, nutrition influences a lot. If you're not getting the proper nutrition, then you're not going to grow to the sort of optimum height that your genetics might be expressing, just because you don't have the raw nutrients and materials necessary to build your body up, if that makes sense.
All right, so genetics can sometimes go in a bad direction. You can end up with inherited diseases as well. Here's an example of sickle cell anemia, which is an inherited disease. Basically, there's a gene that codes for the formation of the hemoglobin protein that's inside of all red blood cells. And if that gene is not functional properly, then the hemoglobin doesn't form properly, and then the blood cells turn into these sickle shapes rather than the nice round shapes that can flow really well through your blood vessels. The little sickle shapes will get stuck and build up and cause problems. Okay, so sickle cell anemia is a problem.
All right, there's also something called sex-linked disorders. There are certain genes that are found only on the X and Y chromosomes. All right. With females, we already established this. There are 2 X chromosomes found in every cell. And certain genes other than those that code for your gender can be found on that X chromosome. And sex-linked disorders in general tend to be a recessive trait. So in order for a woman to have a sex-linked disorder, both of the alleles on the X chromosome would have to be messed up and have the disorder gene. All right. With guys, because guys only have one X and one Y, if they get the problem gene on that X chromosome, they're going to have the disorder because they don't have the backup X that women have to kind of, help fix it, So if guys end up with a problem gene on the X, then it's going to get expressed because their other chromosome is a Y. Sorry, guys. That's why sex-linked disorders tend to occur much more frequently in men than in women. Of sex-linked disorders include hemophilia. And hemophilia is a blood clotting disorder. It's caused by a mutant gene that codes for a particular clotting factor, and it prevents the blood from clotting properly. So if you get even small cuts, you can virtually bleed to death. And it's found on the X chromosome. Okay, so males who inherit this defective gene will be unable to produce the clotting factor properly and therefore will have these bleeding problems. In heterozygous females, so if a female has a problem gene on one X but has a normal gene on the other X, the normal gene will produce enough of that clotting factor so that the girl is able to clot properly and prevent themselves from bleeding out. But because they're actually have that bad gene or that bad allele on that one chromosome, they're considered to be carriers. Okay, they don't show symptoms, but they're able to pass that allele on to half of their sons. And so they can actually cause somebody else to have the disease. All right, so they're known as carriers. Women rarely will suffer from hemophilia because in order for them to actually have hemophilia, they have to inherit a defective gene from their father as well as their mother. So they'd have to get both defective genes. And until fairly recently, there were very few hemophiliac boys that got, basically grew up old enough to have children. They would die when they were very young. So women were always carriers and men were the only ones who officially got it. Nowadays with, current medical advancements, it's, it happens a little more often that, it happens a lot more often. Men are able to grow up and actually be old enough to have children. And so it's possible for women, more probable to have for women to have it now than it was before, but still low, right? And if you look at this, women has, as a carrier here, normal male. You have 25% chance of having a normal female, 25% chance of having a normal male, 25% chance of having a carrier female, and 25% chance of having a male who has hemophilia. Okay, this male would have hemophilia. If you cross a heterozygous woman with a man who has hemophilia, you would get a girl carrier, a normal boy, a girl who has the disease, and a boy who has the disease. Okay, as probable or possible children. All right. There are some traits that are found only on the Y chromosome. And one of them is kind of a funny one. It's hairy ears. And so it's found solely on the Y chromosome. Males are the only ones affected. And because it's only on the Y chromosomes, if a guy has a son, the Y chromosome comes from him, so the son will always have hairy ears as well. So if any of you out there are wondering if you're going to end up with this, you need to look at your dad. There you go. All right, I just wanted to mention what a pedigree chart was. I think we brought these up briefly before. They're similar to family trees, but they're used to trace a particular trait across generations of a family. We use squares to indicate males. We use circles to indicate females. And there's a horizontal line that will join a circle and a square to indicate that a couple had children. And then there'll be a branch that goes below that to show what children they had. In general, a fully shaded in trait or fully shaded in shape will indicate that they have that trait that we're looking at. And a half shaded in shape will indicate that they are a carrier for that trait. Okay, here's an example. This is a pedigree for hemophilia in the royal family because it was a problem in the royal family. Somewhere way back, we know as far back as we know, for instance, that Queen Victoria was a carrier because she had a son, Leopold, who had hemophilia. Okay. We also know that she had a daughter that had to have been a carrier because that daughter ended up having a son that had hemophilia. There's a couple of lines that got, away from the hemophiliac problem. This group in the German royal house ended up without hemophilia. There's another group basically down here which leads to our current royals in England, Queen Elizabeth II, Prince Philip, Prince Charles, their two kids. Everybody over here is okay. The most famous royals that had this problem were, of course, this little cluster right here. Tsar Nicholas and Tsarina Alexandra. Tsarina Alexandra was the granddaughter of Queen Victoria, and she was a carrier, and she passed that trait on to Alexis, her son, the young one. And so he had problems when he was quite young, before, of course, they were all executed during the Russian Revolution. So there you go. A close-up if you wanted to see it, that shows Alice, which is the daughter of Queen Victoria, mother of Alexandra, married to Nicholas, and then there was also some problems over here in Prussia with hemophilia. There you go. A little brief interlude on genetic counseling. Genetic counseling is offered to families who have a history of genetic illness in the family or genetic problems in the family. And genetic counselors can evaluate a family's history and their medical records, and they can order genetic tests, and they can sort of evaluate the genetics of the family to determine what the probabilities are of a particular pairing, passing on a rather destructive genetic trait to their children. So it kind of, some parents actually make the decision to not have children based on the fact that their genes are a certain way that they could give their child a really debilitating, awful genetic disorder. You do it basically by analyzing small samples of blood or body tissues, and then you can determine whether you're carrying certain genetic alleles that for inherited disorders that could be bad for your children. Some familiar genetic disorders, or some that maybe aren't that familiar, but some are like Down syndrome, cystic fibrosis, sickle cell disease, and your book's going to talk about sickle cell disease actually, and we'll do some stuff on Friday on that. Tay-Sachs disease, or the next day. Spina bifida. If you want more information about any of these, feel free to research and look on your own, you guys. There's a lot out there. Okay, we're going to end this with a discussion on selective breeding. Selective breeding occurs when specific characteristics are sought after by either, a breeder, say for dogs or horses or something, or somebody in agriculture for harvesting higher yields on crops, things like that. But it's a way to increase the frequency of a particular trait in the population or a particular characteristic. Okay, and it's how we get basically something as big as this and something as little as this with the same genetics of a dog, because this is the same species. Okay, they're just different breeds. All right. So suppose you wanted a cow that produced a lot of milk. This is what, farmers basically did. They choose the cows in the herd that produce the most milk. They only let those cows reproduce. They select the offspring in the group that produce the most milk, and we only let those reproduce. And you keep doing that over and over again until basically you have a little herd of cows that all produce lots of milk. And people have been doing this for thousands of years. Okay? It's used in agriculture. Over here on the left, this is sort of wild corn, and selective breeding of picking the ones that always produce the most kernels kept happening over and over again until we have the corn cobs that we have today, basically. It happens in the pet trade. Some people think there are certain color combinations in snakes and fish and, oh, apologies to anybody who's afraid of snakes. Sorry. And it also occurs with birds. You just selectively breed for particular color patterns that people think are interesting, and then they will buy them. It happens with cats. This is an early Angora type breed of cat, Persian cat, and over the years you'll notice that the little snout gets flatter and flatter and flatter, because somebody somewhere thought that was cute, which, to me, just personally, this actually causes a lot of problems for cats. Some of these cats, their heads are so flat they have trouble eating. They have eye problems, all sorts of things. So, I think selective breeding can go a little too far at times. Look at that. What is that? Come on. That's ridiculous. But here's some other examples. Wild mustard gave us all these varieties of kale, broccoli, cabbage, kohlrabi. Okay? You can start to see some of the resemblances in these different breeds right here of dog. They are all related. You got a very large cow there. Okay, you get the point. There's some more research out there, but I'm not going to worry about playing this right now. We can talk more about this on the next day, okay? And I believe that that will be the end.